The simple phylogenetic tree constructed on the previous slide, however, does not take into account the significance of individual codon positions. It turns out that a change at the first codon position has a different impact on protein structure modification than a change at the second position, whereas changes at the third position generally have the smallest effect. That's why transversions and transitions are often presented separately for each codon position. Let's take a look at the genetic code table. Let's see what happens if we make an arbitrary transition and transversion at each position. Let's start with transversions, which generally have a greater impact on causing a radical change in the amino acid. For example, a transversion at the first position of the leucine codon TTA to ATA will change this amino acid to methionine. However, a transition at the first position of this codon from TTA to CTA turns out to cause no change in the amino acid. CTA still codes for leucine, but in most cases both a transversion and transition at the first codon position will lead to a change in the amino acid. Similarly, for the second position, for example the same leucine, codon TTA, from our example, a transversion to TAA will change it to a stop codon, which will most likely cause a lethal change, meaning it will completely prevent the correct reading of the entire gene frame. When a transition occurs at the second position of the TTA codon, from our example, changing it to the TCA, the amino acid changes from leucine to serine. In the case of the third position, a substitution will generally have the smallest effect. All transitions at the third position will not affect the encoded amino acid. For example, our leucine codon TTA, after a transition at the third position, becomes TTG, which still codes for leucine. This is true for any amino acid and even for stop codons. According to the table, nothing should change after a transition at the third position. It is slightly different in the case of a transversion at the third position, but here the change will have no effect on 8 amino acids. By the way, the mitochondrial DNA code table can be divided into 24 coding regions, where for 8 amino acids the coding spans 4 adjacent codons, while for the remaining amino acids and 4 stop codons, the coding spans 2 adjacent codons. Let us now examine the distribution of the substitutions for the first position, the second position, and the third position separately. During the analysis, we will refer to the phylogenetic tree constructed earlier. For the first position, we see that the number of transitions between the gorilla and the human and the chimpanzee is set at roughly the same level, around 220. Substitutions between the orangutan and the gorilla, chimpanzee and human remain at the constant value of about 330. Similarly, substitutions between the gibbon and the orangutan, gorilla, chimpanzee and human are at a comparable level, roughly around 330. Substitutions between bovine and primates are approximately 420, whereas substitutions between mouse and the other mammals are approximately 410. The number of transversions between chimpanzee and human is 18. Between gorilla and human and chimpanzee, we also have the same level, around 20. Between orangutan and gorilla, chimpanzee and human, it is around 90. Between gibbon and the other primates, it is about 100. Between bovine and the primates, it is about 400. And transversions between mouse and the other primates are around 500. A very similar distribution can be seen in the table for the second position, although the values of these substitutions are smaller. To explain the effect of transitions and transversions at each position, I took the liberty of calculating biochemical distance coefficients for a single transition or a single transversion for each position in the mitochondrial DNA. Without going into the method of calculating this coefficient, it indicates that the higher its value, the greater the impact a substitution will have on altering the properties and shape of a protein. We can see that the coefficient value for the transversions are 1.41 for position 1, 2.12 for position 2, and 0.37 for position 3. So, as we might expect, the number of transversions will be smallest for the second position, since its impact on changes in protein structure is the greatest. Next, the values are higher at the first position and the highest at the third position. Similarly, for transitions, the coefficient value for the first, second, and third positions are 1.2, 1.4, and 0 respectively, which means we will observe the fewest transitions at the second position, followed by the first position, and the most at the third position. We remember that transitions at the third position do not affect amino acid changes at all. Since we see that the first and second positions have the greatest impact on the changes on protein properties and structure, the substitution table is presented in the form of two separate tables, as the summed value of substitutions at the first and second position and as a separate table for substitutions at the third position. What, however, conclusions can we draw from these tables? Namely, in the table for the first position, we can observe a continuous increase in the level of transitions, all the way up to the transitions for bovine and then for mouse. We can see that for the mouse, there was probably some saturation with transitions, and perhaps from that point on the number of transitions may start to decrease. We observe a slight decrease in transitions for the mouse, probably caused by transitions at the similar sites in the first position, which at a later stage were replaced by transversions. The number of transversions in the mouse is higher than in bovine, and this is certainly a topic worth investigating. However, it does not appear that some saturation occurs for transitions at the second position. For the third position, on the other hand, we observe a huge number of transitions with clear saturation. This value rises to about 1000 for the gibbon, then decreases and remains at a similar level of around 800 for both the mouse and bovine. This may be caused by a large increase in transversions at the third position, which simply occupy sites previously taken by transitions. What is very interesting is that there are numerous transitions at the third position, even though, as we mentioned earlier, transitions at the third position have no effect on changing the amino acid or the stop codon. Similarly, transversions at the third position have a small effect on changing the amino acid and the stop codon. Although they occur in large number, most of them do not cause changes in the protein. Since we observe no effect from transitions and only a minor effect from transversions at the third position on amino acid changes, this suggests that substitutions at the third position and probably at the other positions as well may occur randomly rather than in a directed manner. Importantly, we see that transitions at the third position between humans and chimpanzees occur in smaller numbers than for the other primates, which suggests precisely such a process of random changes associated with the shorter time elapsed since the split between humans and chimpanzees.